Hello everyone, welcome back to our plant propagation courses. And the topic today is micropropagation of some important plants. And the following slide will contain about example of micropropagation used in commercial plants. And until now, for food crops, the example of the use of micropropagation in a large scale is rare, can be considered as rare. Uh, I read that some of the researchers they turn their attention to soybeans and but not start to do propagation in large scale. And for horticulture plants, because of the high value of horticulture products, of course, the use of micropropagation will be very useful. And there are so many examples of the use of micropropagation for horticulture plants, such as in fruits. One of the best example for sure is banana because it's already wide applicated, and strawberry and citrus. And then for vegetable, there's potato, sorry. And then for ornamental plant, there are orchid, gerbera, anthurium, chrysanthemum, etc. And for medicinal plant, this is ginger, or in Indonesia we call it jahe and mint. And this is also very important. Estate crops. For today, I will give you example about the palm oil situation. First is banana micropropagation procedures. Of course, you're already familiar with this procedure because in practicum and also from previous weeks, we already discussed a little bit about this. I just want to refresh you. Yeah. So for banana micropropagation procedures, uh, this is very interesting because the banana plantation now become very huge in the world, and if we only use sucker for uh, plant material, of course, it's not be will not be enough. And I will also tell you about why we should in vitro propagation here. So just to remind you that first we need to choose the appropriate explant, and this is an example. We already give enough explanation to you before. So it should be a buds that have just sprouted, young sucker, and healthy, and the maximum height is about 1 meters. And after that, we should prepare the media, and the media for banana is not really complex. It's just a base media MS with the addition of some plant growth regulator here, and sometimes antioxidants can also be applied here because banana is full of pathogen sometimes. So there's sometimes you can see internal contamination, so plant enhancer will be also useful in the initial stage of the plant materials. So you already familiar with this, and we already show you the video of this process. So from this material, we peel one by one, and then we make a small cut, and after that we use chemical treatment to yeah, to try to eliminate the pathogen, yeah. And after that we transfer it in a smaller dish like this, but before we put it in the sodium hypochlorite, yeah, or natrium hypochlorite for several time using different concentration and times. And after that, we drain it, and then we put it in the media like this, and we wait for this initial material or explant to be regenerated. And after some times, we should explore the explant. If it is contamination, we can do the re-sterilize steps, but you can also just throw it away if it's already like this, it's full of contaminant, and it's very hard to make a cut of it without having some pathogen to stick with the materials. And um, if the explant form callus or new shoots, we can continue to the multiplication media. And multiplication, there's also some plant regulator that can be added to the base media of MS, and we put in the culture room for 26 to 30 degrees Celsius with the photo period of 16 hours light. And we can do subculture into the new media every 4 to 6 weeks, and it's depend on your program and also the certification, of course, for doing this. And in general, that if the buds are already too high, then we should transfer it into the routing. Sorry, if the buds are already too high, we can also do a subculture. But when the plant already high, yeah, enough, then we can transfer it to the routing media. And this is the composition of routing media. I will not talk deep into this. This is just a give you example. And after this banana, we will see that in the other plants, they put different plant regulator, even the different MS concentration. It's just give you an idea about that. And also the periods of routing, multiplication also will be different. And we should do hardening before acclimatization, right? And in the banana case, we do it in the room temperature. And during the acclimatization, as I told you before, so we can use a media with a sterile compost or coco peat, and we can do it individually, or we can do it in a group with a plastic container, styrofoam with holes in the bottom, and cover it with plastic. Or in the large scale, we can use acclimatization bands and use sand, for instance, for the media. And the general procedure is that we should remove the plantlet from the bottle and then wash it using clean water and to remove all the agars. And sometimes we soak it with benlet and plantomycin, drain it, and plant it in the media, cover it for once into 2 weeks until the plant already survive like this. So from this bottle, we put it in the media, I put plastic cover to keep the RH, and after they can already survive like this, we can move the plant to other place, putting a new media, or even you can also transfer it into other place, and also you can put it in the new polybag like this afterwards. So this picture is from last year from the student. We prepare the culture like this, and then the student continue with to do the acclimatization and maintains the plant until this height. And after that, we transfer it into our field station in Sukamantri, and hopefully this year is already give fruits for us. And as a tradition, the use of in vitro in banana is not only for micropropagation purposes, but before that, the in vitro is also interesting because it can eliminate the virus. You can see here for some report, the banana perhaps the most world endangered fruits. Why? Because the massive attack of this Banshee top virus and also Fusarium. Fusarium is relatively easy to be removed uh, using in vitro culture, but for virus, it's a different story. So for virus, we should do thermotherapy, chemotherapy, and meristem culture in banana, and this is the scheme. So from in vivo plant like this, we can take the part like what we did during the sterilization. But before that, we can do thermotherapy using some level of high temperature. Of course, it's already adjusted that the plant will not be too suffered from that condition. And using also a chemical treatment, yeah, using the disinfection of the meristematic zone, and also meristem excision. It's just very small, maybe only 1.2 millimeters. And the meristem excision can also be done after we got the in vitro plant. So basically, after the sterilization, we just remove the, let's say, the fungi and bacteria, but the virus still there. But it's okay. You can get the plantlet, but afterwards we can do the meristem excision to get the plant free from a virus. And after that, we do chemotherapy, and then if the ELISA cannot detect the presence of the virus, we can directly do regeneration of the plantlet to the medium of regeneration. And after several months of subculture, we can get the greenhouse acclimatization from that plantlet. So this is the example. This is the small banana because of the infection of the virus. This is not normal banana, of course. So you can imagine that the virus is systemic. So the virus is also here, yeah, and also infected all the part of the plant materials. So we took a very little, let's say, a meristematic part here, and see, this is only six centimeter, and we peel it and we cut it into only 2 centimeter, let's say, and we do exactly the same with what we previously already discussed. We do the sterilization, but after that, we will cut the meristem. So the meristem is diagrammatically look like this. So we can cut, let's say, 1 millimeter part of the meristem tip from, let's say, this example is the in vitro rooted plantlet. So we take from the in vitro like this, and then we cut it, the very small part of it, and uh, again we do the kind of sterilization. And this is the picture. We need the binocular, of course, to do this, because here, for instance, we cut the parts just 1 millimeter big like this. And as I said before, we only need perhaps 1.2 millimeter. So maybe only this uh, small part. Okay, so for banana, I hope it's already clear for you. So next, the plant that are also very popular for to be propagated using in vitro is potato. So from uh, variety of genotype, the researcher do uh, selection and also crossing to get the desired characteristic of the new variety. So I did this in the field and with hotter temperature, because I want to get the potato with good adaptability to higher temperature conditions, such as in Indonesia. So after that, we conclude with, for instance, this variety. And before dissemination, we need to do the propagation of the propagules. And this is involve the in vitro technique intensively. So after we got the seed, and then we disseminate to the farmer, so the farmers can produce the potato with our variety, and fortunately they can get the increase of their uh, production. So the process of producing potato seeds from this uh, tuber, yeah, you can get the bud here, and then after that we do also acclimatization using this part, and we can get the plantlet like this. And in potato it's relatively easy because it's very easy to get a new shoot like this, yeah, with a very dense node. And after that we can do acclimatization, transfer it in the fully protected greenhouse to get uh, G0, or in Indonesia we call it G0, and we can continue to produce the G1 and the G2 to the G1 produced in the greenhouse but is not really protected, and G2 usually we produce it in the field. Yeah, so this is the scheme, yeah, for making a potato seed, and maybe I can continue also to this. So the use of in vitro is not only to produce the plantlet using nodal cutting, but before that, the in vitro is very interesting in potato because it can also eliminate the viral disease that very big problem in potato production. So we can get potato tuber still from this kind of plant, and after that we can get the buds like this, and we do the meristem culture like what I already explained to you in the banana situation, something like that. And we can also do the thermal application or chemical application, and then after that we get the new plants, and we before we do the nodal cutting propagation in vitro, we should do the ELISA test to see whether the plant containing virus or not, because we expect that we can produce a virus-free material. So after we get the free disease material, we continue with the multiplication. In potato, it's very popular because it's very easy. We can use another culture like this. So from this part, we only use a single node culture, and we put it in the media, and we use a medium MS, just a normal MS medium, using photoperiod 16 hours light and 8 dark, for instance. But in my experience, I can also use a 24 hours light, and the temperature should be around 20 to 22 degrees Celsius. And after that, let's say this is maybe only 1 month, 4 weeks, something like that, you can get already a lot of new shoot. So beside of producing plantlet in vitro propagation situation of potato, we can also produce microtuber, which also desired by farmer because it's comparing to that of cutting or plantlet. This propagule is easier to be transported and storage. However, the price is still become a problem because it's not really economic. But still, some people are still using it. Uh, how can we produce it? Is that after doing multiplication in this media for let's say 4 to 6 weeks, we can add liquid media containing half of MS and sugar in this concentration. We should also use a retardant such as coumarin and kinetin, and then we pour it in the media because it's a liquid media. You can see here that this is the solid media, and here you can see we put a liquid media, and we put it in the photoperiod 24 hours in the dark, so 24 hours without light, and we can keep it in the temperature of 20 to 22 degrees Celsius. And after, let's say, 4 to 6 weeks, you can get this microchamber already formed. And using micropropagation technique, in the end, we can transfer the propagule in the fields in three possible ways. So using in vitro microtuber production, and then mini tuber production, it means that the plantlets should be acclimated, and then we plant it in the media, and the plantlet will produce a small tuber called mini tuber, like this. So this is not very big. So this is the, yeah, it's the control. This is a pen, and this size is only like this. Yeah, and uh, beside that, we can also use the cutting itself to be transferred in the field. So this is the process of acclimatization. So we put the plantlet in the media like this, and after that, the plant will be adapted and produce roots, and it will grow, and it grow like this. Yeah, it will produce yeah, the node like this. Yeah, and we can do cutting ex vivo, so it's not in vitro again. So in the greenhouse, we can also make a cutting like this using single or two nodes. Usually for the beginner, we use two nodes, but the expert of some expert farmer, they use only one or single node. And then after that, we can get a very beautiful plant like this, and this is already ready to be go directly to the field for G2 production, or we can plant it in the greenhouse for the production of G0. So we planned it. So this is the situation of the cutting in the screen house, and after that we transfer it to the poly bag or pot, and from one plant you can get a mini tuber like this, and this already give uh, a good business, because one canal or one tuber like this for G0, it can cost you about 2,500 rupiah, and we can also produce it. We can also use it for G2 production continue in the field, and we can also use the result of the plant acclimatization or the cutting to the aeroponic system. We can get also the bulb from this aeroponic system. Yeah, so this is a bare or naked roots of the potato in the dark condition like this, and we give mist containing nutrition, so the plant can produce mini tuber for that from that. Okay, so enough for potato. We continue to the ornamental plants. One of the famous micropropagation for ornamental plant is orchid. Yeah, I will tell you a story about this. Orchid, based on the type of the growth, can be divided into monopodial and sympodial. Monopodial can be seen that from its main growing point is already is it or only have one growing point. It's not very rare to has side shoot or tiller. Therefore, this is the example. Therefore, the propagation using vegetative conventional way is not that easy. And the second is sympodial. The orchid that is sympodial having more than one main growing point for grow or side shoot like this. Yeah. So to make you remember, this is the sympodial. Yeah, have more than one. See, this already I can already see three or maybe more. But for monopodial, this is only one growing point without has side shoot or tiller. So in conventional way, yeah, of course, the propagation of the sympodial is easier compared to the monopodial. Therefore, the price of uh, this type of orchid usually higher compared to the sympodial type. So for sympodial type, for instance, this is we can do the separation of the new shadow bulb, or we can cut the structure called in keiki or root shoot that form on flower stem or stems. Okay, so propagation of orchid can also be generative from the seed, but the seed of the orchid is a naked embryo or embryo without endosperms, so it cannot be germinated easily. Yeah, so in the nature, orchid seed germinate with the help of certain fungi, and in our case today, we will discuss about artificial seed germination by sowing them in a tissue culture media. So when we get the fruit of uh, the orchid like this, it's containing some seed, and then we can pour it in the media, and after several periods, yeah, it will grow into protochrome. Yeah. So protochrome can form secondary protochromes, and we call it protochrome like bodies or PLB, and develop into plantlet. And for vegetative propagation, for instance, this is for Phalaenopsis because uh, uh, yeah, we can also. So we can also use this shoot culture or using auxiliary branching for orchid for producing uh, new plantlet, yeah, for propagation material. But of course, comparing to the generative ways, like what I've told you before, this is the multiplication, the sorry, the number of plant that you can get in one generation is very much less. But we do not expect that one shoot will produce one shoot, because from one shoot containing meristematic cell, we can induce the protochrome like bodies also from the vegetative tissue, like such as leaf pieces. And uh, how do we do that? We taking young shoots from the parent tree, and then we remove the leaf and do the explant sterilization, and then blend it in the liquid media, and then after that we induce the protochrome like bodies, multiplication of the protochrome like bodies, and root shoot and root induction in solid media, and then after that we got the PLB that are ready to be acclimatized, and then we do acclimatization in the greenhouse. And this picture show you just the illustration of how we do that. So we got the keiki here, and after that we cut the young part of that because it's containing meristematic tissues or cell, and then after that we can produce the PLB, yeah, and we can separate the PLB into new media, and then we can get a lot of new plants. And this is the example of the development of the PLB. Yeah, this is from 3 weeks, and then after that we can produce even the flowers. But of course after acclimatization. So once the PLB are big enough, we can do the acclimatization. Usually we use sphagnum moss media like this, and after that we put it in the uh, greenhouse like this. This is in Indonesia situation, yeah, if I'm not mistaken. So this is Phalaenopsis, yeah, and this is also Dendrobium. For certain kind of Dendrobium that is that has a high price, of course, this the technique also will be very interesting to be applied. And for the medicinal plants, this is ginger micropropagation, and the principle is similar. We can cut the part of the plant containing a new shoot like this, huh? And after that we do the sterilization, and we put it in the right media, and then they will grow producing new shoot. Then after that we can do division, and we can produce new plants. See, uh, using several addition of the plant growth regulator, of course, we can also direct how the plant will be look like, such as this. This is using uh, uh, 4.5 milligram per liter of BIP, yeah, and this is using another types of auxin. You can see that they perform differently, and also with the routing. So it's depend on the availability of planlet and also your program. And after that, we can do acclimatization, yeah, and we can also make optimization of the multiplication and routing, yeah, using sucrose, using auxin, etc. And even for ginger, we can also considering about using micro and mini rhizome. So the system are more or less similar with the potato. So micro means that we can produce a rhizome from the plantlet condition, and mini is that after acclimatization we plant the plantlet, the small plantlet, and then it will give you a mini rhizome like this. And then next is palm oil micropropagation. This is very interesting. Why? Because the palm oil production in Indonesia is very huge, and area is not just 1,000 hectares, but it's already a million hectares. So you can imagine that in some period they need to replant it, so they need to have a new plant from new reliable source every years, let's say. uh, so the need of the new plant is high, and the problem is that variety they use is hybrid. It's called Tenera from Dura, cross with Piscifera. Why? Because in the 100% Tenera, they can get more profit compared to combination of Tenera, Dura, and Piscifera. And even when they only plant Dura, they will uh, get loss in profit. So they prefer to have all the plants in their area is Tenera for production. And you may know that during the hybrid, when we use the seed for propagation, you will get segregation. Okay, and so this is clear that we should produce the hybrid seed. And if you compare to the let's say pepper, yeah, or tomato, is very different, because it's not that easy to produce a hybrid seed from the seed of the uh, palm oil. So to solve this problem is that we use a clonal propagation of really proven Tenera variety. But we face the problem that the oil palm tree architecture naturally is only built up from a single stem without branches. It means that's not too many apical meristems there, and uh, uh, it only has a one apical meristem even located below a cylinder in immature leaves, and uh, from that we can already think that it's not that easy to do vegetative propagation here. So we need to do in vitro culture to isolate the apical meristem here, for instance, or from other parts. And also for consideration, when we cut this apical meristem, so the plant will be suffer, and I don't think it will be survive. Yeah, so that's the problem. Therefore, the oil palm is typically propagated in vitro by indirect somatic embryogenesis. I hope you still remember about that. It's a process in which somatic cells of an explant of choice, yeah, via an intermediate phase of callus growth induced to differentiate uh, into somatic embryos. Yeah. The architecture of the oil palm is lacking axillary shoots, does not allow for vegetative propagation. I think I already explained it before. Therefore, somatic embryogenesis is the only alternative to seed propagation in palm oil condition. So, for somatic embryogenesis protocol, some commercial and also researchers they use immature zygotic embryo, mature zygotic embryo, immature male inflorescence, immature female inflorescence, immature leaf, and young plantlet, of course, from their collection. So this is the step of indirect somatic embryogenesis. So this is an immature of zygotic embryo, and they cultivated into producing callus, yeah. And after that, they develop into embryonic callus, and this is not a uh, fast work because this is usually it's almost 1 year to produce or to regenerate one plant like this. And after that, the embryogenic cells transfer to a culture medium, and in the end, it can regenerate into plant. And after that, they can plant it individually like this. And after that, of course, they do the acclimatization before in the nursery, and they do they put it outside in the field for uh, producing uh, material for replanting. And it's not that easy. So here we identify the problems associated with the oil palm somatic embryogenesis. First is a limited availability of explant. You can imagine we should cut the plant, but that's not desirable for company. So they use instead for commercial production they use immature leaf. It means that leaf that have not emerged yet. So uh, they think that it's also containing a lot of meristematic cell. The constraint that is that it can only be sampled once every 3 to 5 years because the leaf of the oil palm is not that intensive like other plants, right? And then low efficiency of somatic embryogenesis is only around 5% of the explant that regenerates. Yeah, so the company facing this problem, so they still have to do a lot of works to solve this problem. And I forgot to mention, for instance, like what I know in Sinarmas biotechnology in Sentul Bogor, they regularly do this kind of thing, and they harvest the immature leaf from their side plantation in Rio, for instance, and they ship it in the Bogor, and they start to initiate the embryogenesis. And to go via proliferation of embryogenic structure, because it needs a long time and the material will be exposed to chemical and also artificial light, for instance. So the risk of somatic clonal variation is also high. It can lead to the abnormality. Yeah. Okay, next is sugarcane micropropagation. I just give you a brief introduction of that. Yeah, so in the estate crops, sugarcane is also uh, one commodity, also commodity that also use micropropagation technique for making new plant materials. See, like this picture, are basically the same. Yeah, uh, but this is maybe easier compared to palm oil. See, and after that they do acclimatization and transfer it into the field. And another popular example is gerbera. Gerbera is a high value commodity, but it takes difficulties to do the propagation vegetatively because the nodes are in naturally the nodes are very tight, so it's not that easy to separate the or to do vegetative propagation. So they induce it in vitro, and they can uh, produce a new shoot of that, and then they can separate it like this compared to the natural condition. It's easier, and they can produce a lot of new plants. Strawberry, yeah, is also popular for its use, and uh, yeah, it just give you an idea about that. Yeah, so from the runner part of the strawberry, yeah, we can isolate them and then do sterilization, and after that you can get the uh, shoot proliferation, and then you can do multiplication, separation in the new or individual container. You grow or induce the root system, and then after that you can transfer it into the ex vivo condition. Anthurium is also important. It's not in maybe not in Indonesia, but I put here because in Europe, for instance, this is very extensively done, and they do not use a shoot. They induce the somatic embryogenesis. Yeah, because in Anthurium, yeah, you can search from the literature if you're interested. The situation is almost like Gerbera. It's not that easy to get the uh, new plant from one cluster, but yeah, uh, and in other part the demand of anthurium is really high, so they decide to use a somatic embryogenesis here. So they produce callus, yeah, and after that they propagate the callus, and after that they induce into the new plant, yeah, and they can do multiplication here, yeah, and they can already transfer it into the ex vivo condition by acclimatization, and then put it in the new pot. And this is a very huge business in Europe, for instance. Okay, thank you very much for your attention. This just the beginning for to lead our discussion. So thank you very much for your attention.